Chapter 19 talks about molecular genetic analysis and biotechnology. Molecular genetic analysis involves recombinant DNA technology, which are a set of molecular techniques which do the following functions: localize a gene, isolate it, alter it, and ultimately analyze it. These set of techniques can be used for any organism and are particularly useful in the area of genetic engineering for gene targeting, repair, and editing. In order to use recombinant DNA technology, we must first cut and join DNA fragments. In other words, manipulate the DNA fragments. First of all, we're going to cut them, and we're going to cut them with restriction endonucleases, which cut DNA at specific fragments. These restriction endonucleases were originally obtained from bacteria. There were approximately 800 enzymes for 100 sites. There are two types of restrictions endonucleases used. HIND3, obtained from Haemophilus influenza, which is going to cut at the AAG sequence in the 5' to 3' sense, or GAA sequence at a 3', 5' sense. And what's remaining will be known as the sticky end. And we also have SMA1, obtained from Serratia marcescens, which will cut at sequence CCC or GGG in the 5', 3' sense, or GGG CCC at the 3', 5' sense, and what's remaining will be known as the blunt end. After the DNA fragments are cut at specific points, we're going to covalently link double-stranded DNA in different ways by the use of DNA ligase and ATP. These DNA fragments are going to be loaded into an agarose, which is polysaccharides from seaweed or jello. And this is going to go gel electrophoresis, gel being the agarose, in which the electrophoresis for electric currents, in which the negatively charged smaller fragments are going to run faster and are going to be observable under UV light. So once we have all this, how can we examine genes and gene products? Well, sometimes we might have to amplify the DNA. And amplification of DNA is done through PCR. There is a formula for PCR in which the number of copies of DNA is multiplied times 2 to the n representing the number of cycles. And PCR uses thermostable DNA polymerases, which work at 3 stages: denaturing at a 90 to 95 degrees Celsius, annealing at a 52 to 60 degrees Celsius, and amplifying at a 70 to 74 degrees Celsius. But what if we have RNA? There is a PCR for RNA, exclusively for RNA, but this is going to be reverse transcriptase coupled PCR, in which the RNA is going to react with a reverse transcriptase that's going to produce a complementary DNA, its complementary DNA strand, and this will be what's going to be studied in the PCR. So PCR requires DNA. If you have RNA, you need to use reverse transcriptases in order to obtain the complementary strand. The example for this is COVID-19. COVID-19 is an RNA virus, which has traces of RNA in mucus, tissue, saliva, and body fluids. The RNA obtained from the samples of these tissues is going to react with a reverse transcriptase, generating the complementary DNA strand, And this is what's going to be studied in the PCR. We also have that fragments of DNA, RNA, and protein can be studied with the southern, northern, or western blot respectively. A southern blot studies specific DNA fragments. And first of all, we're going to have to extract the DNA from the cells, cut the DNA with the endonucleases, transfer the DNA blot to a membrane from the gel, then use a DNA probe which will bind to target DNA and hybridize the specific gene, and we're going to detect the signals for that specific gene. Next, we have northern blot, which targets specific mRNA, and we're going to extract the RNA from the cell, run the electrophoresis on the gel, transfer blot to membrane, immerse with membrane with label probe, and detect the signals that mRNA is going to give. Lastly, we have western blot for proteins. We're going to extract the proteins from the cells, run a page or polyacrylamide gel electrophoresis, transfer the blot with the proteins onto a membrane, immerse the membrane with specific antibodies, and detect signals or visible bands. Since these antibodies are going to react with the proteins, there is going to be a phenomenon called chemiillumination. The chemical reactions are going to produce light. So we can study two parts of the gene library, the gene itself or how the gene is expressed. In order to study a gene itself, we're going to extract the DNA from the cells, cut pieces with restriction enzymes, join the fragments, and insert them into a plasmid, which is going to be a vector. We're going to transform a colony of bacteria with this plasmid or vector, and this new bacterial population is going to contain the genomic sequence that we would like to study. But what if we want to study gene expression? Well, studying gene expression involves studying transcription, and transcription depends on when, where, and how genes are transcribed. So in order to study gene expression, we're going to extract the mRNA from the cells, convert it to the complementary DNA strand, put this complementary DNA strand into the vector or plasmid, inject the bacteria with the plasmid, and study the bacteria population containing the complementary DNA. But what if I would like to know where the gene is located? Well, for this, I'm going to use FISH, fluorescent in situ hybridization, which is going to allow us to determine the distribution of specific mRNA in tissues, since it's going to label the DNA or RNA with a radioisotope or fluorescent tag, which is going to allow us to study the gene in those tissues. Two other things that we can study from DNA are the RFLP and the microsatellites. RFLP has restriction fragment length polymorphisms, or changes of length of DNA fragments that were created by the restriction endonucleases, and the microsatellites are variation in the number of short tandem repeats found in centromeres and telomeres. And the study of this is used to generate genetic markers to analyze variation, disease, genetic relationship, linkage, DNA profiling, and genome-wide disease analysis. CRISPR technology, which is a sort of technology that edits DNA in deletion, insertion, replacement, or modification, is also of particular interest. And this was once first observed because it conveyed bacterial immunity to target and destroy invading viral DNA. Another example is the TMC1 gene responsible for deafness in mice, and how editing this gene allows the subsequent generations to not be born deaf, while leaving the gene as is still creates deafness. And out of all of this, we have transgenics. From the CRISPR technology, we also have something similar, akin, which is transgenics, which is the introduction of a known DNA sequence into an organism. And this creates transgenic animals, different effects to virus infections, and this can be done through microinjections. All of these genetic studies can be approached in two ways: forward approach, which is a more traditional approach and it involves just studying the phenotype responsible for the gene, or a reverse approach in which the genotype is studied first and then the phenotype is observed and the gene is mutated in order to find the function of the specific gene. Lastly, biotechnology has great application in genetically engineered bacteria, plants, animals, cells, food, gene testing, and therapy to meet with the demands of modern day life. Okay, that's all for chapter 19.